We do have a question. So if you all want to pop your questions in, we do have one question from Joanne asking if there could be some clarification around SARS-CoV-2's requirement for reverse transcription in the central dogma. So I don't know if you want to just sort of like clarify that aspect.
So there's, if I went back to the screen of the, should I go back to?
You can, yeah, if it's helpful, go for it. Mm-hmm.
Okay. Okay, so when it goes from this RNA molecule to this messenger RNA, it uses a what we call is a polymerase, or this is just a fancy word for saying a protein that copies this to another molecule. So it uses it's an polymerase that makes RNA based off RNA. So obviously that's not the way DNA is transferred to mRNA. So for the virus, it's not really a reverse situation, but it's the kind of molecules that our cells use to make reverse transcription. I guess, I don't know if that made any sense.
Yeah,
So,
I think like it's always like tricky because when we're so used to learning everything in these like DNA based systems, right? And so remembering that they're in RNA and they have to go backwards and then forwards and like all of these things, and they're also replicating each other, and like there's a lot of biology going on.
In this case, it's just going forward.
Yeah, and I think there's also the difficulty, since the way that we're doing the testing, for us to be able to work with that, you need to be able to make DNA, and that's where the reverse transcriptase comes in for us. So we have a question from Catherine Soltani, who asks, if the genome sequence can be studied now for COVID-19, or I guess the new coronavirus, can that help us with predicting future coronaviruses?
It may, and so there's even a lot of the research that has been done to study the receptor binding domain has come from research from studying the original coronavirus. So it's not just all this research just happened, but it's actually many years of understanding the structure of the genome. And so if this also mutates further, it could also help us understand if the current coronavirus changes in ways that are important.
Great, thanks. And that was my cousin. Hello, Katie. Hope you're well. Okay, so we have a question from Facebook from Peggy. Do you know if there have been mutations observed since the outbreak began in humans?
So there have been some mutations, but those mutations, so in general, mutations can occur, and viruses can occur mutations, for example, more faster than we can. And so there can be lots of different mutations, and again, it's also about if all the DNA or all the genes or genomes of the viruses are being sequenced properly. But there's two mutations that have occurred in that have kind of been passed along, and that have allowed people to really track different populations. But whether those mutations are affecting people in different ways, that hasn't really been answered.
Cool.
Okay, so question from Lauren Freed, which is, how do scientists know to look for the virus in bats and pangolins? Is there a reason behind why bats carry so many coronaviruses?
I think so. One of the things to think about is that viruses are just all around us. So it's one of the most abundant types of genetic material, and like living being or almost living being that there is. So there's even viruses that infect viruses. And we may have other kinds of viruses that aren't infecting us so much. But it's just, I guess, known from prior studies that that bats host a lot of these coronaviruses. Probably after the initial outbreaks, it was a race to find. But also that's part of this collecting different materials from different animals. Some people, even during times when there's not an outbreak, they're just collecting lots of viruses from different animals. The pangolin actually had symptoms that were very like what coronavirus is now, and so it was probably just a standard procedure that they kept the tissues and the cells of the pangolin, and I'm not sure if they sequenced the gene right away, but maybe once they saw the similarities, then they may have.
Yeah, thanks. And I think also I know that there are bat people who are very specifically studying coronaviruses in bats, so they're more like bat ecologists. And then there's virology people who are studying coronaviruses who are now also, or who have been for many years, talking about bats as a reservoir for other things. So this is not something that, I just wanted to also add that is not something that is new. Everybody in the scientific communities have known that coronaviruses are these things that are existing in bats for a really long time. And so there is quite a bit of literature on coronaviruses in bats all over the place. So I'm gonna ask a Facebook question, another Facebook question from Carolina. And the question is around how we can easily explain to others what a lab signature is, maybe in a simple way. And just following up on that, how easy would it be to create a virus like this in the lab? And I also want to preface this question with just saying viruses have been around for billions of years. Humans have not. So they're incredibly sophisticated. So I'll just throw it at you now.
Yeah, and so what I mean by lab signature is they're usually specific genes that people use that make things resistant to different drugs, or allow us to see them under a microscope, and they, like, for example, turn green. And so those allow us to really look at things, or like otherwise just kind of enrich for them to make them actually studyable. Otherwise they'll be kind of mixed up with all the other viruses. And so I don't know if that clarified, or if
yeah, we call them like little selection tags, in the lab. So I don't know if it just helps to make sure that they remain concentrated.
Yeah, it's like also being able to stick a label on something and then be able to pick it out at a later date.
Yeah.
And so like, for example, now this coronavirus, the new coronavirus, has been taken and put into those kinds of sequences so that people can study them and really figure out how they work, and so that we can create drugs and stuff like that. So it's not an impossible thing to do, but we would expect that if this virus was out there infecting people, that we would be able to see that signature from the sequences of patients that had coronavirus.
Yeah. And do you think it's easy to create something like this in a lab?
Well, the thing is, yeah, we don't really know. Like, nature has a lot of opportunities, and we have a lot less kind of foresight to know actually what's going to be all the things that can create a very, very deadly virus. Currently, we know now that this virus is very deadly, but that's only in hindsight.
Yeah. So I feel like Aditi and I are becoming firm friends. I always ask her questions. So she has a two-parter, and this is there a specific mechanism for pangolins or bats to transmit the virus to humans? And how would this be different from human to human transmission?
Well, I don't think there's a specific, anything different than humans transferring it from. It's just the way that people interact with these animals. So, for example, the pangolin that was found, these don't naturally live in China. They're found in different areas of the world, and they're usually trafficked animals. And so, just not having that kind of interaction with that pangolin would have helped if it wasn't trafficked. The bats obviously are not, or I don't know if they're how people are interacting with bats, but it's just if you're how you behave or interact with things in close proximity, right?
So this is a really great question from Tiffany, and that is around the implication that SARS-CoV-2 is 96% related to the bat sequences that were examined here, yet the receptor binding domain is more similar to the pangolin.
Mm-hmm.
What does that mean?
So I guess one possibility is that so the way I showed how the genes of the virus get transmitted, it happens like in this very stepwise fashion. It's like one mutation, next mutation, but also sometimes the strains of different viruses can mix together. So different regions and mutations can then transfer. So it could be possible that a coronavirus like the one that is seen in the bat also mixed with the one that's found in the pangolin. And another possibility is that there was already one like the one that we saw now, but it just wasn't sequenced.
So we have a quick question from Joanne, which is this virus present in pangolin saliva?
I'm not sure.
Yeah, I was thinking just in case this was something that was mentioned in the course of writing this paper, like how the animals were sampled for the to be able to detect the virus.
Right. Yeah, I think they just took the tissues from. Actually, it was 2 dead pangolins, and they had died from coronavirus-like symptoms, and so yeah, I don't know how they took the sample from.
So I think there is a question that is asking around, like, maybe are there other animals that could have also contributed to this whole thing? And so, like, why did the scientists focus only on the bats and the pangolins? Were there other things, or maybe are there other things that are not even in our range of thought or understanding?
So the reason why they focus on it is because they found similarity from those sequences that were in that catalog. And so the way this worked is actually they didn't even sequence the whole genome of the viruses. So they just sequenced a specific region of a specific protein that the virus has. And they first assess like how similar in that region, and then they actually have to do a lot of sequencing. So it's actually quite a lot of work because it's very difficult to sequence the whole genome. And so there are probably different animals out there with very with coronavirus-like symptoms, but these are the ones that were in the catalog. So for example, I think we know that cats can also they also have an ACE2 receptor that is very similar to humans, and that allows for the new coronavirus to bind to that. And I'm sure there are other animals. Camels were initially affected in previous coronavirus outbreaks.
Okay, so from Aditi again, is the rate of mutations in pangolins and bat coronaviruses parallel to that of humans?
So the rate of the coronavirus should depend on the actual coronavirus. And it actually has some proofreading when it's replicating. It has a proofreading way to correct itself. So its rate of mutation is lower than other viruses that usually don't have that proofreading ability. But I wouldn't imagine it depending on the host, but yeah.
So we have a great question from Anissa around ACE2. And so like, what is it normally, if you're aware of what ACE2 normally does in our bodies, and then, maybe like, why ACE2? Why would the coronavirus use that as the receptor? If you even know, I don't even know if we know that answer.
Yeah, so I don't know what ACE2 is doing on the cell. So I know that it's a receptor and that it's expressed on the cells of lungs, but other tissues as well. But I don't think it's the it's not like looking to take advantage of the ACE2 receptor. It's that the ACE2 receptor has some structure that allows it to kind of enter cells and infect cells and continue being able to have its life cycle. So if it can't infect cells through interacting with the ACE2 receptor, then it has to find a different way to keep living.
So I had one question before we tap out, I guess. And this is more regarding where the information for the paper came from. So given that this is a review and the people presenting the information didn't do the experiments, how did the authors choose the papers that they then subsequently describe?
So the way authors usually do this is that they review the articles within the field, and they they use which is most relevant to the kind of case that they're trying to make, and they also read the papers very thoroughly and try to establish which one is more credible. Yeah, it's mostly them looking and actually developing this knowledge over a long period of time of knowing what is the research out there and what is everybody saying. So we have kind of different ways of sharing information, from different meetings or just reading articles, and so that's also why 7 different people wrote this publication. So people have different expertises that may overlap, but there's going to be things that some people are more expert in.
So we have a bunch of scientists right now on the panel, and I'd like to maybe open it up to everybody if anybody wants to join in. But I was just wondering, like, what are your thoughts on the pace of research and how we've been sharing this information? Like, I personally feel that I've never seen science move so fast in my life. I feel like we're in like this like Ferris Bueller situation that if we don't stop and look around, everything will pass us by. And I don't know, maybe Ferris Bueller is a bit of a tell for my generation. I don't even know if you know who Ferris Bueller is out there. But and do you think that this will change the way we think about doing science in the future and how we think about collaboration and things like that?
All right. Is anybody else gonna?
I don't know. Does anybody else want to join, or does anybody have any thoughts on that? Or you can start us off if you want, Andrés.
I think science is quite a collaborative endeavor already. There are some competition. I mean, there's quite a lot of competition, but there's also this willingness to share as well. that's increased a lot. So there's a lot of people doing things and sharing everything more openly and trying to kind of solve this problem together. But you know, if it's not related to the pandemic, I'm not sure if that kind of feeling is going to continue, or
Right.
you know, but
so at some point I was working in a lab doing Alzheimer's research, and it feels like for a very long time this has already been something that, as you say, has been going at breakneck speed. And I think that's partly due to the scientific structure, the fact that scientists are expected to publish so many papers that for Alzheimer's, there is just no earthly way you could read every relevant paper that came out on a given day. So it would be interesting to see how this moves forward, because I think that potentially we don't need more information. We just need it to be more succinct and easier to translate. And I know some people are going through journal articles and they're trying to compile, you know, the most relevant bits of information from certain papers. And I don't know how you do that without losing any of the information. But yeah, I wonder if the course of COVID-19, everything that's coming out as a result of it will help us hone the amount of information coming out.
Yeah, I think, yeah, I think all of, I think your point, Andres, also on like, will this apply to non-COVID stuff? I don't know. That's a really fair point. And so thanks for sharing that.